Hello, and welcome to Badger Talks Quick Picks, which bring exciting happenings, resources, and talent from your UW flagship campus to the people of Wisconsin and beyond. My name is Mike Bertram. I grew up on a dairy farm in Fond du Lac County and have been with the Agricultural Research Station since graduating from UW-Madison with a degree in agronomy. I started in Spooner, then Marshfield, and I've been managing the Arlington station since 2012. Today, I'll give an overview of the Arlington Ag Research Station, including a brief history, followed by an introduction to the different types of research being conducted around the station. The Arlington Station was established in 1955. At the time, research was taking place at the Hill Farms, which were on the west side of the UW campus. The farms were purchased by the UW in the late 1890s and were a great place for them to do the research. However, after World War II, the soldiers were coming back and attending UW-Madison. The city was growing as well as the campus was growing, and the 600 acres to the west of campus weren't going to be in research for long. The leaders of the university at the time were able to negotiate and purchase 13 farms in the Arlington area, which amounted to about 1,800 acres. And in 1955, operations were moved up to this area. The area is known as the Empire Prairie, and it is a good Plano silt loam, great for growing crops on. This farm expanded through the following decades, adding 200 acres in the 1960s, and then the last addition was in 2022, which was 70 more acres, which was added specifically for organic research. So what do we do here in Arlington? Our main purpose is to support researchers from campus. So we do field operations support for the crops researchers. That includes tillage, planting, pesticide and fertilizer application, and help with harvest in some cases. We also support the animal researchers with growing the forages, the bedding, applying the manure, and doing different repairs. We also mix and deliver research feed for the animal researchers. We truck equipment and different things around for those who need it. We have a couple of mechanics who do routine maintenance and repair for equipment and vehicles. And we also, because there's a lot of interesting things going on, we fabricate and modify equipment as well for doing research. And then finally, we have a lot of facilities, different buildings, and we have a crew that repairs, remodels, and does different projects to keep facilities operating. Another interesting thing we do have is a public events facility so that we can host different events, field days, and outreach so that we can share these results with the public. On the crop side, we have 2,000 acres of cropland. About 1,750 of that is owned. The rest is rented from local farmers. About half of that land is devoted to crop research, and about half is used for raising feed for the research livestock units. The main crop we have is corn with about 800 acres. We have about 400 acres of alfalfa and other forages, 350 acres of soybeans, 150 acres of small grains. And then for every other crops, we grow just about anything else that can be imagined. We generally have about 70 different crops growing on the station each year, which can include things like sweet corn and other vegetables, switchgrass, different types of grass, kernza, sunflowers, many more things. One of the newer ones is industrial hemp that several researchers have been working with. We also have land in pastures and land in tree research. The station has about 92 acres of certified organic land and 64 acres being transitioned. Some of the different departments we work with: plant and agro-ecosystem sciences. They do hybrid trials, different crop management things, weed control, long-term crop rotations, plant breeding, and alternative crops and uses. Soil and environmental science works with nutrient management of grain and forage crops, manure management, nutrient cycling, soil management and water quality, and different work with cover crops. Plant pathology and entomology are departments that work with plant disease and insect control in different crops that are studied. insect monitoring collection. One of the new interesting things is called the insect eavesdropper, where it uses microphones in the field to detect if insects are present and how much they are based on what it hears. Different pollinators and biological control for insect pests. Biological Systems Engineering Department does a lot of neat things with equipment. Some of it involves biofuel crop harvest and transport, different ways to harvest and store forages, manure storage and management, some environmental work looking at runoff and other things, how crops and manure affect the environment, egg safety and health, and then a lot of work with precision agriculture, sensors, GPS, RTK, things like that. On the animal side, the swine unit has about 1,500 hogs. It was originally opened in 1985. They do all life aspects of the hogs, from gestation, farrowing, nursery, and finishing. It is a biosecure site, which means that everyone who works there showers in and showers out. And because it's biosecure, the pigs grow more efficiently because they're not fighting diseases. Some of the different research focused on swine genetics, reproduction, nutrition, and management. And another interesting aspect is the swine unit has some pigs that are high cholesterol, and they're used for biomedical research. The sheep unit has 2 main barns. They can house up to 300 ewes and plus their lambs. They also spend some time on pasture, a lot of the summer. A lot of the research is toward improving production and genetics and reproductive techniques. So they use some programs such as semen collection and storage, artificial insemination, ultrasound, and embryo transfer. They also have a big teaching and extension component to the sheep, and many are exhibited and consigned at state and national sales. And then finally, our dairy facility. It's a state-of-the-art sand-bedded freestyle dairy facility. It was opened in 2008. Some of the research includes nutrition work, mammary and reproductive research, calf growth studies, transition management. They can do individual intake studies, and recent research has to do with animal comfort and welfare. They can milk up to 490 cows in this facility, have 100 dry cows, and 60 calves. Next, we'll hear from Laura Hernandez about some of the important work being done at the research station. Laura's research is focused on the coordination of physiology during pregnancy and lactation. Her work is centered on improving health, physiology, metabolism, and well-being of moms. Laura will speak on how she has been focused on using mammary ultrasound scanning as a method to determine the ability of the gland to produce milk. By initiating this study in dairy cows, she has been able to follow the animals since birth and is currently monitoring a cohort through most of their second lactation. This data set will not only be important for dairy cows, but will allow her to work collaborators in human medicine to perform pilot studies in women. The ability to ultrasound a pregnant woman and have some clue of how much milk they may or may not be able to produce will allow them to develop breastfeeding plans prior to parturition.
Thank you, Mike, for the introduction. I'm excited to talk to you today about what we're doing in our lab and have been working on for the last several years. These are pictures of the dairy calves up at the Arlington Research Station over the course of several seasons, of which we have been working with these animals as they grow throughout their life cycle. Ultrasound is a really commonly used tool in the healthcare setting, and in women, they use this to identify tumors or breast lumps typically. And we got to thinking, could we use an ultrasound to identify the mammary gland's ability to make milk? And could we do this and transfer this knowledge to the human population so that we're able to help moms determine their ability to make milk prior to giving birth? So we began to use the cow as a developmental model. We're able to follow them through a life cycle, take tissues in order to verify structures, and also apply some other techniques like using 3D imaging in collaboration with our collaborator, Dr. Joao Dorea in the department, And so we got to thinking we could do this, and we're able to use dairy cows because they have a very unique productive life cycle. These animals, and because we keep them in our herd from birth throughout their lactation and potentially several lactations, we can follow them from birth all the way through several lactations and identify structures in their mammary gland and hopefully determine whether they're able to produce maximal or minimal amounts of milk by using the ultrasound and correlating this to corresponding features from histological images. And so because these animals are impregnated at varying points throughout their life in order to make milk, we'd be able to look at these animals not only during their lactation, but also their pregnancies. This is Dr. Alicia Vang, my postdoc and previous grad student who's been doing this study now since 2020. She's been wholly dedicated, had never touched a cow in her life, and has really made use of not only her care and diligence, but commitment to the project in order to improve the lives of women. This is an image of her collecting ultrasounds and also performing mammary gland biopsies. And what's neat about this is we're able to take an ultrasound image and take our biopsy tissue from the exact location within where our ultrasound images are being collected. So we're able to parallel these tissues together. This is just a picture of her securing tissue from the gland. This is Alicia, and you can see here we're able to go behind the rear of the cow and take images from the very top to the very bottom of the gland. And so this allows us to capture the parenchymal tissue, which is where the milk secreting tissue is, the cistern, which is the region where the mammary gland collects milk in a cow, all the way down to the teat. She's been able to do this at Arlington, at Marshfield, at the Dairy Cattle Center, and has made use of all of the very wonderful agricultural research sites we have access to on this campus. And because we track our animals very closely, we're able to follow them closely across time. So you can see here, this is an image in which we have on the left a black structure, which looks like a hole, and a bunch of white tissue. And what we have here on the right is a pink area where we're able to identify where the actual mammary fat pad or the tissue where the mammary gland is growing its secretory tissue is located and the mass next to it. And we're able to identify regions in order to align this with our histological images, making use of artificial intelligence as well as computer vision systems. This is one of our cows, or beginning as a calf, Emily. We've been able to follow her through the course of her entire growth period, all the way till the birth of her first calf. And she has since now had her second calf. And so we've been able to see what happens in a first lactation versus a second lactation and how the mammary growth corresponds. You can see here images at varying stages across a lactation cycle or a productive cycle, 10 weeks of age. You can see here the 6 months of age and how the mammary gland is growing into the tissue and how this gets larger and larger as the animal ages. And these are the corresponding histological images to that ultrasound image where the pink arrows are. And you can see how the adipose tissue is growing, how the luminal and the ductal structures grow. And this helps us identify that the tissue we're collecting is in a region of which we predict on the ultrasound to be the location where the mammary gland is going to be making milk. These are images of varying stages of life from calf all the way up to a pregnant animal of biopsy tissue we're able to collect from the mammary gland of these cows. And you can see here how we're able to stitch these images together using our microscopes and histology and able to identify by hand where these luminal structures are. This takes an enormous amount of time. And when we zoom in, you can see these structures even closer, where we can identify the adipose tissue and the secretory tissue. And then we can also see the connective tissue, which is where no lactation would occur. And using AI and segmentation, we're able to identify these structures and make use of AI technology to build a model and a tool that would allow us to make this high throughput, because doing this takes about 8 hours per tissue, an enormous amount of time. And so these AI technologies allow us to take a look at the raw image and overlay where we would expect the lactational structures to be versus the non-lactational structures. And this will be foundational for us to identify how much a mammary gland might make milk in a pregnant animal or a pregnant woman, which is our goal. And so this AI technology would allow us to do this in minutes versus hours. But we have to develop the ground truth before we develop these models. And since we've been inputting all these images and we have thousands and thousands of images inputted, we're up to about a 98% accuracy of the AI tool to the ground truth, which is really exciting for us, not only as a science lab, but in the development of this technology for pregnant and lactating women, with the goal that we can actually help women know how much milk they might produce so they're better prepared when they give birth, to know if they're going to reach sufficiency during their lactation. Because believe it or not, anywhere from 15 to 40% of women are unable to reach sufficiency, or in other words, be able to feed their baby at the amount to not supplement with formula. And so if we can make a woman's life better, we would be really thrilled to do this. And really, this research station and the access we have to these animals not only helps agriculture, but it allows us to transfer our knowledge to human health. And this data set and more data sets we continue to collect are allowing us to form relationships with folks in the medical school in order to take this into practice. And now, 5 years later, we're really excited. We're very close to this. And every day we get a little bit closer, and we wouldn't be able to do it without the Ag Research Station. And so with that, I show you how we pull all this data together. We hope to be able to leverage these images and utilize our technology in collaboration with not only my lab, which is the group and the researchers that do the work, all the grad students and postdocs who help us get this done, but my collaborator, Joao Dorea's lab, who also, without their knowledge in technology and artificial intelligence and computer vision, this wouldn't be a reality. That combined with the animals that we have access to and the people that work at these farms, we are really thankful to move this work forward. Thank you.